Welcome to part 2 of the lecture series dealing with macromolecules. What we discussed in the last lecture was an introduction to macromolecules, and briefly what I want to do is review that for you, and that a macromolecule is really just a large or giant scale molecule. Most of the macromolecules are what we consider polymers, which basically means they are long chains of repeating units that are either identical or very similar to one another. Those individual units that are repeated are referred to as the monomer, mono meaning single, single unit being repeated over and over and over again to make the long strand polymer. Now again, this is true for most macromolecules. We also discussed briefly that making and breaking of polymers is a very similar process for the polymers themselves. Synthesis of a polymer occurs via a dehydration reaction or a condensation reaction, and that is going to produce the release of water in that synthesis reaction. Hydrolysis is almost the opposite type of reaction, where water is going to be utilized to break apart covalent bonds to release individual units and break down the polymer itself. Also in our last lecture, we then talked about 2 of the 4 categories of macromolecules. We discussed proteins, and proteins again are extremely diverse type of molecules. What unites or unifies proteins is the fact that they are long stretches of amino acids that are linked one right after the other. And again, we talked quite a bit about protein structure in the previous lecture. We also talked about nucleic acids. Nucleic acids are molecules that are information storage molecules. We discussed the 2 categories or types of nucleic acids, DNA and RNA, and talked quite a bit about the structure of these molecules as well. So what we will do with today's lecture is talk about the 2 additional categories of macromolecules, that is carbohydrates and lipids. Carbohydrates, in a general sense, are fuel and building materials. When we talk about carbohydrates, they include lots of different types of molecules. They can be small sugar molecules, such as things you find in soft drinks. These are monosaccharides and disaccharides. And they can also be very long molecules, hence the polymer component, in things such as pasta and potatoes, and these are what are referred to as polysaccharides. So what we'll do is we'll look at each of those individual categories in the next series of slides. Monosaccharides with the prefix mono indicate single monomeric units. These are what we typically refer to as simple sugars, something like glucose or fructose. Again, these are considered very simple sugars and also known as monosaccharides. These can be the monomer that are utilized in a polymer when we get to that point in talking about carbohydrates. When we talk about monosaccharides, there are some characteristics that we need to discuss. The molecular formula for any of the monosaccharides is going to be some multiple of CH2O, and this is again how the carbohydrates got their name, carb for carbon and hydrate because of the H2O. The monosaccharides will all have a carbonyl, and the carbonyl position determines the classification. So going back to those functional groups that we talked about, the carbonyl is a carbon with a double covalent bond to an oxygen, and that's highlighted in both glucose and fructose in the yellow color. The position of the carbonyl dictates the category or class of molecules that they fall into. We talked about aldehydes and ketones. More specifically for monosaccharides, when we talk about them in the sugar form, they would be either an aldose or a ketose. Monosaccharides can also be classified by their size of the carbon skeleton, and that can be anywhere from 3 carbons to 7 carbons for different types of monosaccharides. One last thing I want to mention with this particular slide, tying in one of the terms that we've talked about in a previous lecture, and that is both glucose and fructose are structural isomers of one another. The molecular formula is exactly the same. However, the way that the atoms are arranged in each one of these molecules is different, in particular the location of the carbonyl, and that makes these molecules in fact different from one another, even though they have all of the same atomic components that make up the molecule itself. Now, because our focus really is biologically relevant molecules, one of the things I want to point out about glucose is that it's not typically a linear structure. In aqueous solutions, many monosaccharides form ring structures like you see here for glucose. The next group in the carbohydrate category is a disaccharide, and a disaccharide is two monosaccharides joined by a covalent linkage that's referred to as a glycosidic bond. Again, this is the name for the bond that links two monosaccharides together. So in this case, you have two glucose molecules that are being linked together to form the disaccharide. This is again a condensation or a dehydration reaction like we reviewed at the beginning of this lecture and we talked about in the previous lecture as well. The most common disaccharide is table sugar or sucrose. Sucrose is a disaccharide formed between the monosaccharides glucose and fructose. Sucrose is extracted typically from sugarcane and the roots of sugar beets, and it is a very common carbohydrate found associated with plants. Now, monosaccharides and disaccharides are considered major nutrients for cells. In a process called cellular respiration, cells can extract the energy that's stored in chemical bonds of a typical monosaccharide molecule. Monosaccharides and disaccharides then tend to be utilized again as a major source of nutrients for the cell in order to extract energy in those chemical bonds. A disaccharide needs to be broken down typically first into individual monosaccharides, but those are quick sources of energy for a given cell. We transition now with this slide to talk about polysaccharides. And polysaccharides are polymers of monosaccharides. So this is where the polymer comes in, or the polymer concept when we talk about macromolecules. Polysaccharides can be hundreds to thousands of units long, and each individual unit or monomer is in fact linked by those glycosidic bonds that I highlighted a few slides back. When we talk about what polysaccharides are utilized for in a typical cell or organism, there are two main functions associated with polysaccharides. Either as a storage molecule to store energy, and I'll get to that in just a minute, or a structural component overall. So in this particular slide, you have three different examples of polysaccharides. The first one is a polysaccharide known as starch. This is a storage molecule for plants in that simple monosaccharides like glucose, such as you see in the top portion of this figure, are linked together one right after the other in order to store that energy-containing molecule. Like I mentioned, the number one source of energy for a cell or nutrients for a cell is a monosaccharide, and it is usually glucose. So if there's excess glucose, it can be stored in these long chains for future use inside the cell or the organism. In panel B, you see glycogen. Glycogen is also a polysaccharide. It is the storage molecule to store glucose inside animal cells. So the way animals and plants store glucose is different in the structural characteristics of the polysaccharide. Regardless, they are long stretches of glucose molecules linked one right after the other. In glycogen, there tends to be branching of the structure, whereas in starch, it is a much more linearized structure. But in the same case, glycogen is a way to store glucose for future use inside an animal cell as an energy source. Lastly, in panel C, you have the molecule known as cellulose. Cellulose is also a polysaccharide, and it is a structural component in plant cell walls. It is a long polymer of glucose. The polymer, however, is in a different structural arrangement than what you see in starch, and this again adds structure and support to the plant cells overall. The last category of macromolecules is lipids. And lipids I always save till the end because this is a category of macromolecules that does not include polymers. So the first three examples we talked about, proteins, nucleic acids, and carbohydrates, all have some polymer component associated with them. Lipids, however, do not have that polymer component associated with them. Lipids are grouped together overall because they have very little or no affinity for water. These are extremely hydrophobic molecules, and it is this hydrophobicity that makes them fall into the category known as lipids. And to quickly review what it means to have a hydrophobic molecule, or what makes a molecule hydrophobic in the first place, is that it is a molecule that is dominated by non-polar covalent bonds. And it's these non-polar covalent bonds that make it, the molecule in question, not readily interact with water. So lipids all fall together into this category because they are extremely hydrophobic molecules. Within the lipid category of macromolecules, there are three subcategories: fats, phospholipids, and steroids. So we'll focus on fats first. Fats are molecules that do in fact store energy. And I know that's not what we typically think of as a fat, but hopefully I will be able to explain that here with the next set of slides. A typical fat molecule is made up of a 3-carbon alcohol known as glycerol, and that's highlighted in the green overall in the top portion of the figure. And it contains 3 fatty acid chains, and a fatty acid chain is a long hydrocarbon-based carboxylic acid molecule, typically 16 to 18 carbons in length. The way that the fatty acid is linked to the glycerol is a dehydration or condensation reaction, and that's what you're seeing in that top portion of the panel. So at the bottom portion of the figure, you have the complete fat, which is the glycerol, and 3 fatty acids linked to it. Fats can be further subdivided into what's known as an unsaturated fatty acid or a saturated fatty acid. The term saturated refers to a fatty acid that has the maximum number of hydrogen atoms associated with the carbons. What this really means is that there are no double covalent bonds in the structure, and what you see in this figure with the first fatty acid chain is a very straight or linear type of structure. So the first fatty acid that you see in this fat molecule is what's referred to as a saturated fatty acid, and that means it is saturated with the maximum number of hydrogens bound to carbon atoms in the structure. And structurally, this makes for a linear molecule. The flip side of that is an unsaturated fatty acid, and an unsaturated fatty acid has less than the maximum number of hydrogens bound to carbons. What this really means is that there is at least one position where there is a double covalent bond between 2 neighboring carbons. If you look at the fatty acid chain that's on the bottom of this fat, you can see the double covalent bond sort of where the bend is in the structure. And this is really important because that bend in the structure causes some very different behaviors and characteristics of the molecule. And we will come back to that a little bit later. So again, with a fat, a typical fat is a glycerol with 3 fatty acid chains, and then unsaturated fats are going to have unsaturated fatty acid chains. That means there's going to be at least one double covalent bond somewhere between 2 neighboring carbons. Saturated fatty acid chains have the maximum number of hydrogens, meaning that there are only single covalent bonds, and structurally these two are very different or distinct. Unsaturated have bends in them or kinks in the chain, whereas saturated fatty acids are very linear or straight molecules. So what's the biological relevance or the difference between the two categories of fats? What you see in this figure is or are some examples of saturated fats and unsaturated fats. Saturated fats are things, again, that have very straight linear structures because there are no double covalent bonds present. And at room temperature, these things tend to be or act as a solid. These are your typical animal fats. Unsaturated fats are going to have at least one double covalent bond, which then puts sort of a kink in this chain, making it not nice and linear, but sort of bent in structure. These are things that are typically more your plant fats. There are some animal examples, like you see in the last column with the omega-3 fats from something like salmon or from shrimp. But in most cases, these tend to be fluid or not solids at room temperature. And these are things that you typically do see with your plant oils that you might cook with. Those are not solids at room temperature, whereas butter is definitely a solid at room temperature. And this has to do, again, with the structural characteristics of the saturation versus the unsaturation. Saturated fats are straight and linear and have the ability to compact really tightly one next to one another. Unsaturated fats have the kinks or the bends, and that allows for more space between neighboring molecules, which allows for a liquid or a semi-solid type of situation at room temperature. The next category of lipid is a phospholipid, and we're going to talk about its structure and its relevance here, and we will review that and come back to it and talk about it more extensively in our next lecture when we begin to discuss membranes associated with cells. Phospholipids themselves are very similar to fats. They are composed of the glycerol molecule as the 3-carbon chain where fatty acids are linked to them. 2 of the carbons do in fact have fatty acid linkages to them, and the 3rd carbon is linked to a phosphate group. So in this figure you can see structurally many representations of a phospholipid. So a phospholipid gets its name because of the phosphate group that is present. What this does, though, is generate a molecule that is very interesting in function. The phosphate component has a net negative charge. Net negatively charged components or molecules interact readily with water. They are hydrophilic. However, the fatty acid tails are made up of hydrocarbons, which are dominated by nonpolar covalent bonds, which makes those portions hydrophobic. That makes a typical phospholipid a very interesting molecule, and that a portion of it is hydrophobic and a portion of it is hydrophilic. And because of that sort of dichotomous nature overall, it makes this molecule interact with other like molecules in a very similar way. If you have a series of phospholipids coming together in an aqueous solution, they will typically form what you see in panel A, which is known as a micelle. This is a 3-dimensional sphere where the hydrophobic tails tuck away from the water and the hydrophilic heads, where the phosphate groups are, interact readily with water. As I said, we're going to come back to this and talk about the structure in much more detail in our next lecture, because this is what allows for biological membranes to form. It is the hydrophilic and hydrophobic portions of the molecule that interact in a specific way to form what we know as the lipid bilayer, which is the structural characteristic of membranes inside cells, outside and inside cells. The last category of lipids are known as steroids. There are hundreds of different types of steroids that are known or have been identified. They typically have very different structure and function from the fats and the fatty acids and the phospholipids that we talked about previously. They typically have a carbon skeleton that is made up of 4 fused rings, and an example of that is what you see in the center of the slide with cholesterol. Hormones are signaling molecules. There are lots of different examples of these signaling molecules. Sex hormones, for example, play a role in differentiation and reproduction. Corticosteroids play a role in maintaining cell function and metabolism. Anabolic steroids play a role in increasing muscle and bone synthesis. So there are lots of different functions for steroids, but lumping them together, they do act as signaling molecules, and they also have this very similar carbon skeleton of 4 fused rings together. By adding different functional groups is what then allows for the production of different types of steroid molecules overall. What you can see hopefully at the bottom is 2 different types of steroids, testosterone, which is a male-based hormone, and estrogen, which is a female-based hormone. And the structure of the molecules are very, very similar. There are subtle differences in functional groups that allow for the differentiation of each of these hormones overall. So that's the tail end then of our lecture series dealing with macromolecules. And hopefully you can go back and review the 4 categories and understand the 4 different categories and why they are so important from a biological point of view.